channels are integral proteins that span the membrane and when open permit the passage of certain ions. It is characterized as selective, may be open or closed, or has a conductance of channel. So this picture is an example of a cell model and shows how cellular gradients and membrane potential are established. On the number 1 example is the sodium-potassium ATPase decreases the intracellular sodium and increases the intracellular potassium. Some potassium exits the cell via the potassium selective channels and generates the action potential. Next is the sodium, glucose, and the sodium amino acids importers use the energy in the sodium electrochemical gradient directed to bring sodium into the cell to drive the secondary active cellular uptake of the glucose and amino acid. Similarly, the inwardly directed sodium gradient drives the secondary active extrusion of hydrogen from the cell, thus contributes to the maintenance of intracellular pH. It only shows that the example on the number 2, the energy in the sodium electrochemical gradient drives the transport of other ions and molecules through and the use of various solute carriers. While on the number 3 example is the action potential drives the chloride out of the cell through the chloride selective channels, thus lowering the intracellular concentration below that of the extracellular fluid. While on the number 4 shows that the 3-sodium calcium antiporter along with the plasma membrane calcium ATPase extrudes the calcium from the cell, thus contributes to the maintenance of a low intracellular calcium. To understand what determines the magnitude of action potential, it is important to recognize that any transporter that transfers the charge across the membrane has the potential to influence the action potential. So you can check my previous video about the membrane transporter for further explanation. Diffusion potential. Diffusion potential is the potential difference generated across a membrane because of a concentration difference of an ion. A diffusion potential can be generated only if the membrane is permeable to the ion. The diffusion potential level across a membrane that exactly opposes the net diffusion of a particular ion through the membrane is called the Nernst potential for that ion. So the picture shows the differences across a selectively permeable membrane. On the figure A, the potassium concentration is great inside a nerve fiber membrane but very low outside the membrane. The electropositivity outside the membrane and the electronegativity inside because of negative ions that remain behind. So the potential difference between the inside and the outside is called the diffusion potential. On the figure B, this is the establishment of a diffusion potential when the nerve fiber membrane is permeable only to sodium ions. Note that the internal membrane potential is negative when the potassium ion diffuse and the positive when sodium ions diffuse because of the opposite concentration gradients of these two ions. Equilibrium potential is the diffusion potential that exactly balances or opposes the tendency for diffusion caused by a concentration difference. At electrochemical equilibrium, the chemical and the electrical driving forces that act on an ion are equal and opposite, and no more net diffusion of the ions occurs. Resting membrane potential. This is the concentration difference of the permeant ions across the cell membrane. It reflects the equilibrium potential of most permeable ions and thus the highest equilibrium potential. Note that all cells have resting membrane potential and only excitable cells have an action potential. And it has negative 70 millivolts. Action potential. It has two refractory periods, the absolute and the relative. For the absolute refractory period, this is the period during which another action potential cannot be elicited, no matter how large the stimulus. The inactivation of gates of the sodium channel are closed when the membrane potential is depolarized. They remain closed until repolarization occurs. No action potential can occur until the inactivation of gates open. Relative refractory period. It begins at the end of the absolute refractory period and continues until the membrane potential returns to the resting level. An action potential can be elicited during this period only if a larger than the usual inward current is provided. The potassium conductance is higher than at rest and the membrane potential is closer to the potassium equilibrium potential, and therefore farther from the threshold. More inward current is required to bring the membrane to the threshold. Depolarization makes the membrane potential less negative. The cell interior becomes less negative. The inward current is the flow of the positive charge into the cell. The inward current depolarizes the membrane potential. Overshoot occurs during an action potential when membrane potential is more than 0 millivolt. Repolarization refers to the change in membrane potential that returns to a negative value just after the depolarization phase of an action potential, which has changed the membrane potential to positive value. Hyperpolarization makes the membrane potential more negative. The cell interior becomes more negative. The outward current is the flow of the positive charge out of the cell. The outward current hyperpolarizes the membrane potential. After hyperpolarization is the hyperpolarizing phase of the action potential where the cell's membrane potential falls below the normal resting membrane. This is also commonly referred to as action potential's undershoot phase. The upsurge of the action potential, which is the inward current, depolarizes the membrane potential to threshold. The depolarization causes rapid opening of the activation gates of the sodium channel. The sodium conductance of the membrane promptly increases. As it becomes higher, the potassium conductance and the membrane potential is driven towards the sodium equilibrium, which is the 65 plus millivolt. Because of this rapid depolarization during the upstroke is called the inward sodium current. The depolarization also closes the inactivation gates of sodium channel, but more slowly than it opens the activation gates. The closure of the inactivation gates results to closure of the sodium channels, and the sodium conductance returns towards to zero. The depolarization slowly opens the potassium channel and increases potassium conductance to even higher level than at rest. The combined effect of the closing of sodium channels and the greater opening of the potassium channels makes the potassium conductance higher than the sodium conductance, and the membrane potential is repolarized. Thus, the repolarization is caused by an outward potassium current. Potassium equilibrium potential has a millivolt of negative 85. Propagation of action potential. It occurs by the spread of local currents to adjacent areas of membrane, which are then depolarized to threshold and generate action potentials. The conduction of velocity is increased by increased fiber size and the myelination. The increased fiber size increases the diameter of the nerve fiber, results to decrease internal resistance. Thus, the conduction of velocity down to the nerve is faster. The myelin acts as an insulator around the nerve axons and increases conduction velocity. The myelinated nerves exhibit saltatory conduction because the action potential can be generated only at the nodes of Ranvier, where there are gaps in the myelin sheet. So here is a picture of a comparison of action potential conduction in an unmyelinated axon and in a myelinated axon. On the figure A and C, at the initial time of an action potential is being generated at the left side of each axon. Note that the inward current in the unmyelinated axon, which is the figure A, is depolarizing adjacent portion, whereas in inward current in the myelinated axon of figure C is depolarizing all of the membrane to the next node. At the second instance in time, the figure B and D, the action potential in the unmyelinated axon of figure B has been generated in the adjacent portion, whereas the action potential in the myelinated axon of figure D has been generated at a subsequent nodes and it is already depolarizing the last node to the right.